Welcome to the lecture on TCA cycle, and TCA cycle stands for tricarboxylic acid cycle. It is also known as the citric acid cycle, and in some books they refer to it as the Kreb cycle after the name of the discoverer, Sir Hans Krebs. Now, before you start looking into the TCA cycle, you should have studied glycolysis, because TCA cycle follows glycolysis in a cell that is pursuing aerobic respiration. That means respiration in the presence of oxygen. Now, in the TCA cycle, we will follow the following roadmap. First, we will look at the transport of pyruvate from the cytosol into the mitochondria. So you should know that TCA cycle occurs inside the mitochondria. If a cell lacks mitochondria, it will not be able to perform TCA cycle. Then we will introduce you to the pyruvate dehydrogenase complex, which is responsible for the conversion of pyruvate to acetyl coenzyme A. We will focus on specific steps of the TCA cycle and focus on a specific enzyme, alpha-ketoglutarate dehydrogenase, and compare it with pyruvate dehydrogenase. Then we will also look at the regulatory steps in the citric acid cycle, and we will conclude the session with the number of ATP molecules that are generated in the process of the citric acid cycle. So, as I mentioned in the first slide, TCA cycle occurs only under aerobic conditions. Now, we have already looked at the conversion of glucose to pyruvate, and under anaerobic situation, pyruvate is converted to lactic acid, or in a cell that lacks mitochondria, pyruvate will be converted to lactic acid. Under situations when the cell is pursuing aerobic respiration, pyruvate goes into the TCA cycle. And in order for the TCA cycle to occur inside the cell, it requires the mitochondria. So if I look at the TCA cycle in brief, the first step is the conversion of pyruvate to acetyl coenzyme A. Of course, specific amino acids also give rise to acetyl coenzyme A, and also fatty acids, after getting metabolized by the process of beta oxidation, also give rise to acetyl coenzyme A. And acetyl coenzyme A then goes into the TCA cycle or the citric acid cycle. So the first point to remember here is that acetyl coenzyme A production occurs in the mitochondria. That means I will not be able to produce acetyl coenzyme A from pyruvate if I lack mitochondria. And all the reactions that are indicated by this black box occur inside the mitochondria. So what is the first step after glycolysis? That means after I have converted glucose to pyruvate, is to transport pyruvate inside the mitochondria from the cytoplasm. And we will have to look at how this process occurs. So to reiterate, if a cell is pursuing anaerobic or anaerobic respiration or respiration in the absence of oxygen, glucose will be converted to pyruvate and pyruvate will be converted to lactic acid. Under aerobic conditions, the pyruvate first has to be transported inside the mitochondria where it is converted to acetyl coenzyme A, and acetyl coenzyme A then goes into the TCA cycle. Now, after it has gone into the TCA cycle, it will produce protonated NADH and FADH2. And sometimes in some books they call NADH and FADH2 high energy electron carriers, and NADH and FADH2 are then oxidized in the respiratory chain, leading to the production of ATP and with the production of water as well. So if someone asks you what is the terminal electron acceptor in glucose metabolism under aerobic situations, the answer is oxygen. Because oxygen takes up the protons from NADH and FADH2 and leads to the production of water. In the process, we also get ATP. The process of oxidation takes place in the respiratory chain, also known as the electron transport chain, which we will look in detail in the next lecture. In this lecture, we focus on the citric acid cycle. But before we go into the citric acid cycle, we need to first transport pyruvate from the cytosol into the mitochondria. So, transport of pyruvate inside the mitochondria happens in two ways. So, the first mechanism is relatively simple. In the first step, pyruvate freely diffuses from the cytoplasm into the mitochondria through channels in the cell membrane, in the mitochondrial membrane, and these channels are transmembrane proteins known as porins. So a simple diffusion, and diffusion means that pyruvate will move across a concentration gradient from a region of high concentration to a region of low concentration. Region of high concentration, that is the cytosol, and the region of low concentration is the mitochondrial matrix. And in doing so, it passes through specific transmembrane proteins present in the mitochondrial membrane, and these transmembrane proteins are known as porins. The second way that pyruvate is transported is by the action of an enzyme known as pyruvate translocase. And the translocase enzyme helps in the transport of pyruvate from the cytosol inside the mitochondrial matrix by symport. And sometimes we call this protonated symport. Symport meaning with each entry of pyruvate, a proton is also transported from the intermembrane space. That means this is the outer membrane, this is the inner membrane, and this is the intermembrane space. And in the process of symport, proton moves from the intermembrane space along with pyruvate by the activity of pyruvate translocase inside the mitochondrial matrix. So, two processes, simple diffusion through porins and symport by pyruvate translocase, helps in the transport of pyruvate from the cytosol into the mitochondrial matrix. Now the question that you need to answer for me is what will happen to glucose metabolism when an inhibitor for pyruvate translocase is added to a typical cell under aerobic conditions. In the exam, when we talk about a typical cell, I designate a cell that has mitochondria and other organelles that you have studied in the cytology lecture of Dr. Nerissa. So what will happen to glucose metabolism when an inhibitor for pyruvate translocase is added to a typical cell that is a cell containing mitochondria under aerobic conditions? Now, once we have transported pyruvate inside the mitochondrial matrix, the first step will be to convert pyruvate to acetyl coenzyme A, and I mentioned about this in the preceding slides. The enzyme complex that catalyzes the conversion of pyruvate to acetyl coenzyme A is the pyruvate dehydrogenase complex. And this pyruvate dehydrogenase complex consists of three separate enzymes, E1, E2, and E3. And the complex requires several cofactors. So what we can say here is that pyruvate dehydrogenase complex consists of three enzymes that act as a module. And this modular enzyme requires the presence of several cofactors for their activity. Now, once we have pyruvate, which is converted to acetyl coenzyme A, a molecule of carbon dioxide will also be produced. So this is the carboxyl group, which by now you already know very well, and the carboxyl group is lost from pyruvate, leading to the production of acetyl coenzyme A. Therefore, this process is called a decarboxylation process. To reiterate, pyruvate dehydrogenase complex that catalyzes the conversion of pyruvate to acetyl coenzyme A is a multi-enzyme complex that functions as a module. When I mention module, what I intend to imply is that each subunit functions in concert with the other and requires the presence of distinct cofactors. So this particular slide is only for your knowledge. You don't need to remember any information from this slide. So if I look at this particular part of the slide, these are the electron micrograph pictures of the pyruvate dehydrogenase complex taken at very high resolution. Now, after taking the picture with electron micrograph, this is the computer generated model of the complex. And as you can see here, the complex acts as a module where three separate enzymes are present, E1, E2, and E3. And this particular star here denotes the binding site for the most important cofactor, lipoic acid. So lipoic acid is one of the essential cofactors that are required, or that is required, for the function of the pyruvate dehydrogenase complex. Now, as I have mentioned here in the slide, this is one of the most important slides, and you need to remember each of the cofactors that is used by each of the enzymes present in the pyruvate dehydrogenase complex. So the E1 enzyme is pyruvate dehydrogenase that uses TPP as a cofactor. The E2 enzyme is a transferase, specifically a transferase known as dihydrolipoamide acetyltransferase that uses lipoic acid or a derivative of lipoic acid called lipoamide and coenzyme A. The third enzyme is an oxidoreductase that is specifically known as dihydrolipoamide dehydrogenase that uses NAD and FAD as cofactor. Now, in an exam, what people will ask you is that they will add a drug in a hypothetical situation which will quench or take away one of these cofactors, and then they will ask you which specific enzyme in the pyruvate dehydrogenase complex will be impaired. So let us say if I take away lipoamide, the transferase enzyme will be impaired and pyruvate will not be converted to acetyl coenzyme A. Similarly, if I take away NAD or FAD, the dihydrolipoamide dehydrogenase will be impaired and pyruvate will not be converted to acetyl coenzyme A. So this slide is very important, and you need to remember the cofactors that are used by the individual enzymes in the pyruvate dehydrogenase complex for their activity. Now, the most important part regarding pyruvate dehydrogenase and the cofactors is related to arsenic poisoning. What arsenic does, it binds to lipoic acid. So if it binds to lipoic acid, and if I go back to the slide here, I will not have any lipoamide. As a result, the transferase enzyme will be impaired. So what happens is arsenic inhibits pyruvate dehydrogenase complex predominantly by impairing the transferase activity of the pyruvate dehydrogenase complex. However, arsenic poisoning also involves the enzyme glyceraldehyde 3-phosphate dehydrogenase. That's a glycolytic enzyme which by now you should be knowing. So what I want you to do here is to go to your book and read the section on mechanism of arsenic poisoning. So if somebody asks you how does arsenic mediate its effect on glucose metabolism, predominantly arsenic impairs the function of the transferase enzyme in the pyruvate dehydrogenase complex by binding to lipoic acid. As a result, the transferase enzyme in the complex cannot function and pyruvate cannot be converted to acetyl coenzyme A. The other side effect of arsenic poisoning is also to impair glyceraldehyde 3-phosphate dehydrogenase, which is an enzyme participating in the glycolytic pathway. So what is the take-home message here is that in arsenic poisoning, carbohydrate metabolism or glucose metabolism precisely is completely impaired, and that leads to toxic effects of arsenic. So in the first step, we have converted pyruvate to acetyl coenzyme A. Once you have acetyl coenzyme A acetyl coenzyme A will enter the citric acid cycle, and the term cycle is very important here. A cycle means I will start at a specific point, go through successive steps, and come back to that point again. So here I start with oxaloacetate and acetyl coenzyme A. In the presence of citrate synthase, it leads to the production of citric acid or citrate. Since the first product of the cycle is citrate or citric acid, I call this pathway the citric acid cycle. Now, once the cycle starts with the generation of citrate through successive steps, it will give rise to oxaloacetate back again. Once we come back to oxaloacetate will bind to further molecules of acetyl coenzyme A, leading to the start of the cycle again. In the process, we produce NADH, that is protonated NAD, FADH2, that is protonated FAD, and also we lead to the production of carbon dioxide at two definite steps. At one of the steps, we produce GTP, which is converted to ATP. So, what are the products of the cycle? The products of the cycle are NADH, FADH2, carbon dioxide, and GTP. But predominantly in the TCA cycle, what we get back is carbon dioxide and protonated NAD and protonated FAD, along with one molecule of GTP. Now, once we have the NADH and we have the FADH2, both these protonated forms will be oxidized in the respiratory chain, where in the presence of oxygen, you will get ATP and water. And NAD and FAD will be regenerated. So now let us look at the first step in a bit of detail. So I have produced acetyl coenzyme A from pyruvate by the catalytic activity of pyruvate dehydrogenase complex. Now you already know that acetyl coenzyme A combines with oxaloacetate, leading to the production of citrate by the catalytic activity of citrate synthase. Please ignore these detailed chemical reactions. Just look at the step. So what you require here to start the cycle is oxaloacetate. So in a cell, if I have oxaloacetate, acetyl coenzyme A will go into the citric acid cycle to form citrate. Now, one molecule of acetyl coenzyme A combines with one molecule of oxaloacetate, giving rise to one molecule of citrate. But let us take a situation where the levels of oxaloacetate are depleted. So I have one molecule of oxaloacetate, but I have 10 molecules of acetyl coenzyme A. So in this process, I will get one molecule of citrate, but the 9 acetyl coenzyme A molecule will be remaining. And what will happen is that these 9 acetyl coenzyme A molecules will lead to the production of ketone bodies. And this particular process happens in fatty acid metabolism because fatty acids produce a lot of acetyl coenzyme A, and in order to metabolize all that acetyl coenzyme A, you require commensurate amount of oxaloacetate, which is not present in the cell. So part of the acetyl coenzyme A will be converted to citrate, but the other remaining part of acetyl coenzyme A will be converted to ketone bodies, and this we will look at later when we study fatty acid metabolism. Therefore, if I do not have enough oxaloacetate commensurate with the number of acetyl coenzyme A molecules, acetyl coenzyme A will be metabolized into ketone bodies. This you should remember. So let us take an MCQ, and this MCQ was present in the assessment in 2017. So a fat-rich diet may lead to increased production of ketone bodies. This is because metabolism of high number of acetyl coenzyme A molecules produced from fatty acids may be limited by the lack of TCA cycle intermediate, and you need to identify the intermediate, and the answer is oxaloacetate, because oxaloacetate combines with acetyl coenzyme A, leading to the formation of citrate, which is the first step of the citric acid cycle. I have given another example of an MCQ related to the TCA cycle. And of course, I'm not going to provide the answer for you here. You need to think about the answer, and you need to identify the correct option. Of course, in order to do so, you need to elaborate the rationale for choosing that option. And please post your answer on the WhatsApp group so that everybody else in the cohort can learn from your reasoning. So the question is, a 54-year-old alcoholic male is admitted to your service with ataxia, ophthalmoplegia, and altered mental status. Which reaction of the cycle below is likely to be impaired in this man? The conversion of pyruvate to acetyl coenzyme A, the conversion of succinate to fumarate, condensation of oxaloacetate and acetyl coenzyme A to citrate, conversion of fumarate to malate, and formation of oxaloacetate from malate. So you need to think a bit on this and come up with the reason for your choice. As I said, we will not focus on all the steps. We will focus on specific steps of the pathway because they are clinically relevant or are important for assessments. So the next step that we will focus on in the cycle is the conversion of isocitrate to alpha-ketoglutarate. So this particular step involves the enzyme Isocitrate Dehydrogenase. Again, I mention here, you do not need to know the structure of any of the molecules. You do not need to know the reaction mechanism involved in the process. What you need to know, of course, is if that process requires a specific cofactor and what is the enzyme or the type of enzyme involved in the process. So in this particular step, we have isocitrate dehydrogenase catalyzing the conversion of isocitrate through successive steps into alpha-ketoglutarate. Since it is a dehydrogenase enzyme, that is, it's an oxidoreductase, it will lead to the release of protons which are taken up by the proton acceptor NAD, and in the process we get NADH plus H plus. For now, ignore the P or nicotinamide adenine dinucleotide phosphate. For the time, ignore this particular entity. We will talk about this in our lecture on pentose phosphate pathway in detail. So the proton that is released is taken up by NAD, and NAD is converted to NADH plus H plus. Also, in this particular step, there is a decarboxylation happening, and this decarboxylation leads to the production of carbon dioxide. So, isocitrate dehydrogenase catalyzed step is an example of oxidative decarboxylation. That means there is oxidation plus decarboxylation. And by this process, isocitrate is converted to alpha-ketoglutarate. The enzyme uses manganese as the cofactor. This you should note down in your notebook and keep a note of it, because if a drug or a toxin specifically removes manganese from the cell, it will impair the activity of the enzyme isocitrate dehydrogenase. So isocitrate dehydrogenase catalyzes the conversion of isocitrate to alpha-ketoglutarate by oxidative decarboxylation. In the process, we get NADH plus H plus as well as carbon dioxide. The next step that we have to look at is the conversion of alpha-ketoglutarate to succinyl coenzyme A. Why we need to look at this particular step is because of the fact that the enzyme alpha-ketoglutarate dehydrogenase is also a complex similar to pyruvate dehydrogenase complex. That means the enzymes in the complex act as a module. The enzyme has the same three or similar three enzymes like the pyruvate dehydrogenase complex, and uses similar cofactors like the pyruvate dehydrogenase complex. So if I want to make an example out of this, in arsenic poisoning, since lipoic acid is not available because arsenic binds to lipoic acid, not only the pyruvate dehydrogenase complex but also the alpha-ketoglutarate dehydrogenase complex will also be inhibited. Now, in the books, we do not mention about alpha-ketoglutarate dehydrogenase complex because of the fact that if pyruvate is not converted to citrate in the presence of acetyl coenzyme A, that's the first step, we will not get this particular step at all, because the intermediates after citrate will not be produced in arsenic poisoning. But for your knowledge, alpha-ketoglutarate dehydrogenase complex is a modular enzyme which has similar cofactors and similar enzymes in the complex like the pyruvate dehydrogenase complex. That's the reason I have written here the effect of arsenic will be the same as you have observed in the pyruvate dehydrogenase complex. Anyway, the alpha-ketoglutarate dehydrogenase complex converts alpha-ketoglutarate to succinyl coenzyme A. In the process, there is a decarboxylation, and also there is the release of protons, which is taken up by NAD, leading to the formation of NADH plus H plus. So since alpha-ketoglutarate is an glutarate dehydrogenase is an oxidoreductase, there is an oxidation and there is a decarboxylation. So alpha-ketoglutarate dehydrogenase complex catalyzes the oxidative decarboxylation of alpha-ketoglutarate to succinyl coenzyme A. The next step is the conversion of succinyl coenzyme A to succinate, and this particular step is important because in this particular step there is a production of GTP. So let me go back. Succinyl coenzyme A is converted to succinate by the enzyme succinyl coenzyme A synthetase. In the process, there is a production of GTP. And this particular step in the citric acid cycle is an example of substrate level phosphorylation. The GTP that is produced in this step is converted to ATP by the enzyme nucleoside diphosphate kinase. And you can see here GTP plus ADP, and there is transfer of one phosphate to ADP. So GTP gets converted to GDP and ADP gets converted to ATP. Now, what I have done is in the LMS, I have uploaded a handout which tells you what substrate level phosphorylation is and how does it differ from oxidative phosphorylation that happens in the electron transport chain. Please read that particular handout carefully because many a times questions are asked where you need to differentiate between substrate level phosphorylation and oxidative phosphorylation. If you do not understand the process, we will discuss it during the in-class session. So, succinyl coenzyme A is converted to succinate by the catalytic activity of succinyl coenzyme A synthetase, leading to the production of GTP, and GTP is ultimately converted to ATP. And this particular step in the TCA cycle or the citric acid cycle or the Kreb cycle is a typical example of substrate level phosphorylation. So in the exam, if I ask you which of the following steps in the TCA cycle or which of the following steps in the process of aerobic metabolism of glucose shows substrate level phosphorylation, you should be able to answer it immediately. Now we have produced succinate, and succinate is then converted to fumarate. Again, in the process, we produce protons which are taken up by FAD, leading to the formation of FADH2. The process is catalyzed by the oxidoreductase, so it has to produce protons which is taken up by a proton acceptor or a proton carrier. Now, When we talked about enzyme kinetics, we talked about competitive inhibition. And I believe I also talked about malonate and succinate. I have highlighted this particular aspect again here because malonate was one of the first competitive inhibitors that is known. And why it is so important? Because it is a substrate analog. That means it shares similar chemical structure like succinate. And that's the reason sometimes questions are asked on this step related to enzyme inhibition. So, which of the following molecules will exhibit competitive inhibition towards succinate dehydrogenase? And you will be given the chemical structures of the molecules. You need to look at the structure, find out the substrate analog, that is the analog for succinate, and the most closest analog that is always given in the options is malonate. So succinate is converted by an oxidoreductase to fumarate. In the process, the proton that is formed is taken up by FAD, which is reduced to FADH2. So we come back to the diagram for the cycle again, and I am not going to focus on the step of fumarate, which is converted to malate, but I will directly go back to the first step, which is the conversion of malate to oxaloacetate. Once oxaloacetate is regenerated in the cycle, it will combine with another molecule of acetyl coenzyme A in the presence of the enzyme citrate synthase, get converted to citrate. So, in order for the cycle to keep on continuing, I need to have oxaloacetate. Of course, I need acetyl coenzyme A as well, but acetyl coenzyme A molecule will be generated not only from the metabolism of glucose, but if you remember the second slide or the third slide in the lecture, it is also generated from specific amino acids and predominantly from fatty acid metabolism. So we have lots and lots of acetyl coenzyme A inside the cell. But in order for acetyl coenzyme A to go into the citric acid cycle, I need oxaloacetate. Therefore, if I inhibit the conversion of malate to oxaloacetate by adding an inhibitor for malate dehydrogenase, acetyl coenzyme A molecule cannot enter the citric acid cycle. And this excess acetyl coenzyme A molecule will therefore get converted to ketone bodies. So that's kind of an example of an MCQ for you. I can give you an inhibitor for malate dehydrogenase and ask you what will be the fate of acetyl coenzyme A under these conditions. And the answer to it is there will be an increased production of ketone bodies. So please remember this. In order for acetyl coenzyme A molecule to go into the citric acid cycle, you require oxaloacetate. If oxaloacetate is absent, acetyl coenzyme A will be converted to ketone bodies. Very, very important. Now we come to the regulation of the TCA cycle. Now I am not going to ask you to read a lot about this, but it is good for you if you can give a casual read through the regulation of TCA cycle from your book. What is the take-home message is that if there is a rise in ADP, that means if there is a fall in ATP inside the cell, so ATP is converted to ADP and PI. So once ATP use increases, the number of ADP molecules inside the cell will go up. So if ADP goes up, then the whole process of citric acid cycle will be upregulated because you need to produce ATP. And there are several enzymes that are affected, and these are the pyruvate dehydrogenase complex and the alpha-ketoglutarate dehydrogenase complex, and also citrate synthase, isocitrate dehydrogenase. So there are several enzymes which are affected by the rise in ADP concentration. Give a casual read through the section related to regulation of TCA cycle from your book. But remember this, that if there is a fall in ATP or if there is a rise in ADP, the citric acid cycle will be upregulated or it will function more. Now, many a times we are asked about the effect of insulin on the citric acid cycle. In order to understand this, we need to first look at the pyruvate dehydrogenase complex. You already know that it's a modular enzyme. You already know that it uses an assortment of cofactors, and it is responsible for the conversion of pyruvate to acetyl coenzyme A. Now, pyruvate dehydrogenase complex exists in two forms: a phosphorylated form, which is inactive, and a dephosphorylated form, which is active. In order for the inactive form to get converted to the active form, we use an enzyme phosphatase, which is a hydrolase which catalyzes the conversion of pyruvate dehydrogenase inactive to pyruvate dehydrogenase active form. And this particular enzyme, phosphatase, is also stimulated by insulin. So insulin activates pyruvate dehydrogenase from its inactive form to its active form. And this pyruvate dehydrogenase then catalyzes the conversion of pyruvate to acetyl coenzyme A. And this acetyl coenzyme A then goes into the citric acid cycle if oxaloacetate is available. So from now on, you need to picturize the 360-degree picture of carbohydrate metabolism. Now, let us say I have enough amount of ATP inside the cell. I do not need to produce ATP. I do not need any further energy. So I need to stop the citric acid cycle. So ATP then activates a particular enzyme, kinase, and this particular kinase converts pyruvate dehydrogenase from its active form to its phosphorylated inactive form, and in the process I produce ADP. So when I have low ATP, I need it to be active. When I have high ATP, I need it to be inactive. Additionally, intermediates such as the acetyl-coenzyme A, NADH also inhibit the pyruvate dehydrogenase complex, because if I have enough acetyl-coenzyme A molecule, I do not need further molecules of acetyl-coenzyme A to be produced. As a result, the pyruvate dehydrogenase complex will be converted from its active form to its phosphorylated inactive form. So this phosphatase and kinase mediated activation and inactivation respectively is similar to what we studied in the lecture related to enzyme regulation. So please go back to those slides, because I mentioned there that these are the general overview by which specific enzymes are regulated, and then translate that particular concept to the regulation of pyruvate dehydrogenase complex. So I have written a small paragraph here. The pyruvate dehydrogenase complex is also regulated by covalent modification. A specific kinase phosphorylates and inactivates pyruvate dehydrogenase, and a phosphatase activates the dehydrogenase by removing the phosphoryl group. So the kinase and the phosphatases are highly regulated enzymes. And I mentioned here what are the conditions that activate the phosphatase and what are the conditions that activate the kinase. Insulin activates the phosphatase, and once the phosphatase is activated, it catalyzes a hydrolysis reaction by which inactive pyruvate dehydrogenase is converted to active pyruvate dehydrogenase. And by doing so, insulin helps in the quick metabolism of glucose, which lowers the concentration of glucose in the blood. So anything that lowers the concentration of glucose in the blood will be favored by insulin. So this is like a concept recall for you, that if you are in an assessment and if you don't remember, you just need to picturize if a particular process will reduce blood glucose. If that particular metabolic process or step reduces blood glucose, you can directly or logically conclude that insulin will be activating or stimulating that particular process or step. Now, I have also uploaded an attachment on the control of the citric acid cycle, and I asked you to read a specific section of the book where you can look at, or where you can read through the general process of regulation of the TCA cycle. But I believe that this particular diagram summarizes the activation or the control of the citric acid cycle better. So if I have increased ATP, increased acetyl-coenzyme A, and increased NADH, the pyruvate dehydrogenase complex is inhibited. Similarly, conditions such as increased ATP, increased NADH will also inhibit isocitrate dehydrogenase, which catalyzes the conversion of isocitrate to alpha-ketoglutarate. Again, if I have increased ATP, increased succinyl coenzyme A, and increased NADH, that means I do not require further ATP molecules to be produced. Alpha-ketoglutarate complex is inhibited, similar to pyruvate dehydrogenase complex, and the conversion of alpha-ketoglutarate to succinyl coenzyme A doesn't happen. So if you look at it again, the pyruvate dehydrogenase complex is inhibited by ATP. The alpha-ketoglutarate dehydrogenase complex is also inhibited by ATP. The pyruvate dehydrogenase complex is inhibited by a coenzyme A, and here it is specifically acetyl coenzyme A, which is the product. In case of alpha-ketoglutarate dehydrogenase, it is also inhibited by a coenzyme A. Of course, in this case, it is succinyl coenzyme A, which is the product. And both the enzymes are inhibited by NADH. So you can look at the functional similarity between alpha-ketoglutarate dehydrogenase and functional similarity between pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase. So they are not only structurally similar, but they are also functionally similar. So the citric acid cycle is regulated primarily by ATP and NADH, and you can see that in the figure. The key control points are enzymes isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase inside the cycle. So once the cycle starts, it is isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase that are the major regulatory points. But the process of carbohydrate metabolism under aerobic conditions also involves the regulation of pyruvate dehydrogenase, and I spent quite a bit of time talking about how pyruvate dehydrogenase is regulated, specifically focusing on the action of insulin on the pyruvate dehydrogenase complex. Now I come to the energetics of glycolysis and citric acid cycle in terms of ATPs. Of course, we don't need to read through the thermodynamics of either glycolysis or citric acid cycle. Remember, the thermodynamics part is not included in the MBRU curriculum, but it is included in some of the licensing exams. So you need to read that section separately. So what we talk about energetics is the energetics in terms of the production of ATP or the use of ATP. So we go back to glycolysis and start from there. We have glucose to glucose 6-phosphate, where we use 1 ATP, fructose to fructose 1,6-bisphosphate, where you use 1 ATP. And then in the glycolytic pathway, we produce 2 NADH molecules because I am producing 2 glyceraldehyde 3-phosphate from 1 glucose molecule. And then I go for 2,1,3-bisphosphoglycerate getting converted to 2-phosphoglycerate, and in this step I also produce 2 ATP because I get 2 molecules of 1,3-bisphosphoglycerate from 1 glucose molecule. Then 2-phosphenol pyruvate to 2 pyruvates, again 2 ATP, and 2 pyruvates to 2 acetyl coenzyme A molecules, which gives rise to 2 NADH. And in the steps of TCA cycle that we talked about today, we get NADH or FADH2. In one of the steps, the conversion of succinyl coenzyme A to succinate, which is the step of substrate level phosphorylation, we get 2 GTP molecules from 1 molecule of glucose and this GTP is converted to 2 molecules of ATP. Now, in our case, what we are going to do is to have it in this particular part. We have done the calculation of the book from where I took this particular table, has done the calculation taking 2.5 ATP molecules per NADH. In your exam purpose, please count this as 3 per NADH molecule and 2 per FADH2 molecule, because this will help you to do the calculation, and the energetics with these numbers does not make a significant difference. So the energetics with these numbers will not make a significant difference. So when you are doing the calculation, what I want you to do is to do this calculation with the figures that I have stated here: 3 ATP molecules per NADH and 2 ATP molecules per FADH2 molecule, and tell me what is the total number of ATP molecules that you are getting. Now, in case of the NADH that is produced in the cytosol in glycolysis, there is a separate calculation, but again I mentioned for our case we will use 3 for 3 ATP molecules per NADH molecule. So what we have done in this lecture, or if I want to recap, we first looked at glucose, which we oxidized to pyruvate. We then looked at the fate of pyruvate under aerobic conditions, where we have now converted pyruvate to acetyl coenzyme A, and this acetyl coenzyme A goes into the TCA cycle, leading to the production of GTP, carbon dioxide, and NADH and FADH2. In the next lecture, we will focus on the electron transport chain, where we will oxidize the NADH and FADH2 that we have produced. And after that, we will look at the conversion of glucose to 5-carbon sugars in the form of ribose 5-phosphate by the process of the pentose phosphate pathway. So that brings us to the end of this lecture slides. Please go through the slides carefully. Read the handouts that have been uploaded, because certain things I believe will be better explained if you read through the notes first, and if you have some queries, questions, or clarifications, we can look at them in the in-class sessions. So thank you very much for listening, and have a nice day.